And now the time has come for the final delightful event of the day. I've heard a lot of people really enjoyed the lightning presentations ahead of posters, Volume 1 yesterday. Now it is time for Volume 2. Something's supposed to happen up there? That's more like it. Okay, take it away, lightning talkers.
McClendon Institute is building a map of neuron activity in the mouse visual cortex.
That's right. You will be able to compare the response of neuron populations to different visual stimuli across many layers and areas.
I need to analyze the data myself. I'm a power user, you know.
All data sets and software tools will be available to download.
I have lots more questions.
You should talk to Wayne Wakeman at the showcase poster session.
So Wayne cheated. My name is Fiona Griffin, and I work with the imaging team. This year, imaging contributed to multiple research programs as listed here. We used bright field and fluorescent microscopy, 2-photon imaging, and intrinsic signal imaging. And this year, we collected about 11,000 1x3 slides, approximately 1,000 3D biosite and field neurons for IVSCC, and collected over 450 functional maps of the visual cortex. So stop by Poster 2 to learn more about the methodology of our high-throughput imaging platforms and see more examples of our work.
Hi, I'm Natalia. I'm from Research Engineering, and my current project is a 2-photon microscope for volumetric fast imaging of clear brain tissue. So we call that scope an isoscope because we aim to achieve equal or isotropic submicron resolution in our data. And this movie represents a small segment of a full coronal section data set acquired on that microscope. If you want to know more about technology, stop by my poster.
Hi, my name is Olga. I'm a part of Synapse Biology Group. Array tomography uses ultra-thin brain sections to provide immunofluorescent imaging with high axial resolution. We propose to increase lateral resolution by using structured illumination microscopy to refine imaging of synaptic structures. If you want to know more, come to poster number 4.
Hello, I'm Dmitry Brewski from Research Engineering, and in this work we demonstrate high-speed amplitude modulation of femtosecond laser pulses, which can be used for multiplexed data acquisition and 2-photon imaging. Among other things, we can use this effect to increase scanning rate of the previously developed random access scanning systems to acquire signals from more than 100 cells in a single millisecond. You're welcome to stop by the poster number 5 to discuss this project in more details. Thank you.
Hi, I'm Peter, also from Research Engineering. We don't just want to listen or look at neural circuits, we want to mess with them. So we're using multi-photon holography to activate small clusters of cells while simultaneously recording the calcium signal as reported by GChem6F in this case. This is a volumetric image where the color represents the depth across 5 slices that span a depth of about 20 microns. The stars indicate where the stimulus is going to hit, and the blinking arrows that you saw show the timing. Please come to poster number 6.
So in vivo deep imaging of cortical layers with synaptic resolution is never trivial because the spatial resolution is always scrubbed by the specimen-induced aberrations. As you can see from those images I obtained at Geneva, we have to use a technique which dynamically measure and correct of those aberrations. With AO correction, we can barely see suspicious spine structures here and there. With AO correction, we can clearly resolve those structures down to layer 5 of the neocortex. If you're interested in knowing more, please drop by our poster. Thank you.
All right, the Allen Institute's new high-throughput data generation pipelines use a lot of different tools and platforms to generate lots of different kinds of data. The big question is how do we keep all of this data aligned? The answer is cross-platform registration. Stop by Poster 8 to find out how your very own manufacturing, process, and engineering team has developed hardware, software, and procedures to keep all of your data internally consistent.
Hi, I'm Jing from Neurocoding, and this movie shows you a simple simulation of mesoscale brain activity in response to a moving circle, based on a retrotopic map generated based on GCAM6 fluorescence wide field image. And if we can train our mouse to play a game like Pong, and I bet that will be how their brain look like. Thank you.
Hi, I'm Josh Siegel from Neural Coding, and the visible spectrum of mice is shifted relative to that of humans. Mice are unable to see longer red wavelengths, but their retina has cones that are highly sensitive to ultraviolet wavelengths, which are invisible to us. However, not very much is known about how the mouse brain processes UV images. So we've taken a three-pronged approach to getting a better understanding of this problem using imaging, ePhys, and behavior. And if you stop by poster number 10, I'll tell you what we found.
Hi, I'm Matt Valley. I'm a scientist in neural coding. In neural coding, we're collecting these amazing movies of the whole dorsal brain surface during behavior, and it's a very rich data set. The first step to understanding these movies, though, is to get a map of the brain from the data itself without having to do anything to the mouse. And so I'm going to present an algorithm that does this in an unbiased and automated way to find brain areas from activity. Thanks.
Hi, my name is Doug Olorensha. I work with Sean Olson in the Neural Coding Group. And what you see on the left is a mouse that's trained to search for and report the presence of visual stimuli on a monitor. And on the right is simultaneously recorded calcium imaging data recorded through the intact skull. And we're using this data to better target future perturbation and single-cell recording studies. If you'd like to learn more or see more of these videos, please stop by my poster. Thanks.
I'm Jennifer Whitesell. I'm a scientist in research here. So we're interested in the default mode network, which is the regions of the brain in the human that are active during quiet introspection. They've also been seen in anesthetized mice. And we wondered, do mice have a state of quiet introspection? And if they do, could we see a default mode network? So I teamed up with Matt Valley from Jack Waters' group, who's doing these behavior imaging experiments of calcium imaging through the skull, and we compared resting state to behaving state imaging. And so come to poster 13 to see if mice have a default mode network.
Hi, I'm Kate Roll. I'm part of the methods development group in structured science, and the poster I'm presenting is on the development of the Intrinsic signal imaging platform in relation to the cortical activity map that you've heard a lot about today. It's poster 15. It's at the top of the stairs, so come check it out.
Hi, I am Dr. ISI. I generate sign maps like this. How come you don't understand? You can see functional regions in mouse, dude.
Very good, Dr. ISI. Now annotate cortical areas in all these signed maps. Good luck.
No! 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 Here comes a turbo auto annotator 2015. Come to see movie logs poster.
And that's poster 16. Thank you.
Hello, my name is Jerome. I'm working on CAM, and so the idea of CAM is to standardize a very important of the kind of system neuroscience experiment we do in neuroscience, where we have an animal behaving, we map grossly the cortex, and then we go in great details to look at the functional response of many cells. If you're still interested in knowing more, you should come first to my poster because I introduce all the different components of CAM.
Hi, I'm Suski Devries, scientist in neural coding. I'm also going to be talking about CAM. We've told you a lot about it today, saying that it's a pipeline to look at visual responses across different cortical areas, cortical layers, and Cre lines to a battery of different visual stimuli. My poster is going to show some of the preliminary data that's been coming off the pipeline, looking across different visual areas, different Cre lines, and different cortical layers. Stop by, I'm at the top of the stairs. This video. It's okay, go ahead anyways.
I'm Pete, poster number 19.
Tom, it's mighty quiet at this CamOps meeting.
Yes, Pete, it is.
Well, where is everyone?
Probably at our amazing poster.
You mean the cross-team pilot study to assess cranial window position and angle in can mice?
Yes. Now it's tickle time.
I'm not ticklish.
Hi there. For almost 100 years, we've spent a lot of time trying to characterize neural activity with electrical recordings such as these. Although recently we've also started doing optical recordings, which produce not only pretty pictures, but also these calcium waveforms, as you might have seen some of those today. Now, the problem is that we'd like to actually relate those two and say, starting with a calcium waveform, figure out when the neuron is spiking. So in order to do that, what we need to do is combine these two methods and provide, do simultaneous OEFIS or UFIS recordings for the German speakers to really characterize the relationship between these two signals. Thank you. Stop by poster 20 if you want to hear more.
Hi, I'm Rylan. I'm a scientist in neural coding, and I'm interested in studying how areas from outside the visual cortex, subcortical areas, signal to the visual cortex to relay information about attention, vigilance, locomotion, and reward. And so the way we're going about doing this is we're expressing GCamp instead of cell bodies, expressing it in axons, and imaging these axons in the visual cortex. And so what I just had on here was an axon, a cholinergic axon in the visual cortex. So come to my poster to see how we're going about doing this.
Hi, I'm Marina. You can come by my poster to learn about the work we're doing, developing a true alternative choice visual discrimination task and combining it with calcium imaging to measure neural responses and selectivity to behaviorally relevant stimuli. And it's poster 23.
Hi, I'm Doris, and we were looking at the go-no-go task in which the mouse was rewarded for stopping at images of birds and mushrooms of different sizes and orientations. The first question we asked was whether the mouse was learning the precise reward contingencies or whether the mouse was generalizing to discriminate the object identity, and we found that the latter was the case. The second question we asked was what was driving behavior, and we found that it was a variety of factors.
Hi, my name's Kathleen. I'm a PhD student in applied math at UW. This poster is about how we plan to use wide field calcium imaging data from mouse cortex to gain insight into dynamical pathways and patterns of information flow. This will result in two products, a set of spatially defined regions and the functional network that they form.
The two-photon serial imaging platform has imaged thousands of whole mice brain using various data products at the Allen Institute. We are moving to a newer and better facility. If you're interested in high-throughput data production, value stream mapping, continuous process improvement, Lean Six Sigma, and how this methodology integrate together to consistently produce this beautiful tissue site data, come see our poster on the second floor.
Hi, I'm Dan. At poster 26, I'll be presenting our work with the IMET consortium in developing silicon probes that are thinner than a single human hair, but have hundreds of individual electrode contacts. At the poster, you'll see some raw data, what it looks like from a brain, and how we can use these probes to record from hundreds of single neurons simultaneously in an awake mouse.
Hello, I'm Sergey. I'm part of the MAT team, where we strive to use extracellular multi-electrode recordings, local field potentials in particular, to help understand the flow of neural activity through the visual circuit. So we use network modeling at the level of biophysically detailed neurons to simulate LFP and the corresponding neural activity in response to LGN inputs. Come to see my poster to find how we put together a network using in-house mouse models, and especially if you have data to inform us.
Hello, my name is Nicholas Kane, and I'm in the MAT team. What is DIP-D? DIP-D is a fast, flexible, open-source simulation environment for coarse-grained neural simulations. It solves a set of coupled partial differential equations for the evolution of the voltage population density equations, providing a statistical description of population-level dynamics. If you didn't catch all that, come by my poster, it's 2.28, where we apply DIP-D to understand the computational properties of a simplified cortical column. It's up near the water cooler, so come by.
Hi, I'm Jung from my team. The in vitro interneurons are extremely diverse, but most of them
express one of the three exclusive genes, PV, SST, and VIP, and we are using the computational models to study their functional roles, and we believe we have some preliminary answers, so if you are interested, please come to poster 29. Thank you.
Hi, I'm Ram, scientist with MAT. One of the questions we are trying to address using in vivo electrophysiological data is what can we know about the dimensionality of the internal brain state between awake and anesthetized mice. If you're interested in knowing more about how we are going about addressing this question using generalized linear models, please stop by my poster 30 on the second floor.
Hello, my name is Séverine Durand. I'm from neural coding. I record extracellular spikes from V1 and LGN and in anesthetized and awake mice. I compare visual properties in these both states. And basically, we found that the main difference is basically temporal. So if you want to know more about it, just come to poster 31, please. Thanks.
Hello, my name is Brian Lee, and I've been working on some of the initial efforts here at the Institute to conduct pair and multi-recordings to assess synaptic connectivity and dynamics. So if you are interested in seeing what I'm doing, please stop by poster 32.
Hi, my name is Brian McClendon, and I work with Shomo in neurocoding. Our ultimate goal for this project is to understand how visual selectivity of cells in mouse V1 emerges from the input of many hundreds of presynaptic cells. The response of a neuron to a stimulus, its measurable function, depends fundamentally on the network in which it lies. But there are very few methods for visualizing such a circuit in vivo. The most promising is a rabies virus that crosses one and only one synapse to label a local connected circuit. Combined will show you how it works.
So information is delivered to neurons through thousands of synapses that form a structural code that through nature and nurture is written into the dendrites and reflected in the wiring diagram. So if you want to continue discussing how we use high throughput electron microscopy to map this wiring diagram and to investigate this code, come to poster 234.
We're moving next generation connectivity forward by going backwards. No, I am not crazy. I am talking retrograde labeling. Yes, we take a modified rabies and we use it to to jump one and only one synapse from Cree-defined starter cells in visual areas. But wait, there's more. Imagine if inducing Cree in selected cells was as easy as flipping on a light switch. It's not magic. We can do that and more. Come check us out.
All right. I should have known this is becoming an annual challenge to follow Ben, I think. One of our many Institute goals is to map long-range connections between visual areas and all the visual-related areas in the brain. And you may ask kind of whether we've already completed this through the previous Mouse Connectivity Atlas project, but the answer we think is no, that we can, as time moves on, new methods become available, and we think we have better ways of generating a more detailed connectivity matrix. And come by our poster to find out what those are.
Hi, I'm Ali from the Neurosurgery and Behavior team, and we have been working on the Next Generation Connectivity Atlas for the past 9 months. It's one of the current pipelines here at the Institute. We're working to perfect several surgical techniques, including ISI-guided viral injections. So come by our poster 37 to hear more.
Hi, I'm Jonathan. I work in the human cell types team, and please come to poster 38 to learn about human ex vivo brain slice recordings. And in particular, I'll share with you some of our feasibility studies at applying genetic labeling strategies, namely virus, to label and study human cortical cell types and circuits. Thank you.
Hi, my name is Tuchman, and along with Basuka Tasic, we will present our recent study in which we characterized and classified cell types in the mouse visual cortex using single-cell RNA-seq. We identified 49 transcriptomic types, 23 of which are inhibitory types, 19 excitatory types, and 7 non-neuronal types. Come by our poster number 39 if you want to learn more about these cell types, including the putative laminar locations, their correspondence to Cree lines, and much more. Thanks.
Hi, my name is Quan Xin. I'm from CCG. Are we all conscious? Yes, we are. So what is consciousness? Consciousness is a state or it's a quality of awareness. So the Christoph and Francis Crick 10 years ago hypothesized that the substrate for consciousness is claustrum. So you want to know more about claustrum consciousness?
Hi, I'm Cameron. So my project starts with a linear model from the first Allen connectome paper and extends it to create a voxel-based model. So instead of finding connection strengths between parcellated regions already, we can create a voxel 100 micron scale connectome. And the first goal is to construct connections within the visual system. And this allows us to probe connectivity between seed voxels that will have different connectivities in this regional voxel model. And we're hoping to use topography of connections to show where retinotopy comes from.
Hi, I'm Carla. I'm part of a team working under the leadership of Julie Harris. And if you're interested in some of the analyses we've been doing using data from the Mouse Connectivity Atlas project, then come see my poster 42.
Hi, Jennifer Whitesell again, continuing our quest to understand the default mode network. This time we're going to look at anatomy. So the default mode network has been defined strictly functionally with functional imaging, but we wondered whether there was a connectivity pattern that underlies this network. So we analyzed the connectivity data on brainconnectome.org. We found some modules that show a high degree of connections, and now we are doing paired anterograde retrograde injections to investigate further the connectivity that might underlie this putative mouse default mode network. Thank you.
All right, thank you, everybody. Can we thank all of the lightning talk presenters again? That was awesome. So before we break for the poster session, I just wanted to take another minute to actually thank all of the presenters, both from yesterday and today. I think we've seen an incredible couple of days of science. I'm just kind of beyond impressed and inspired by the work that's going on, not just that we've heard from our new crop of the next generation leaders, but also internally. I think it's really amazing what we've done in this last year, even from the previous showcase. So thank you, and another round of applause for all the presenters too. Finally, I just want to say that our progress is only really made possible through our founders, Paul G. Allen and his sister Jody Allen. So we also want to thank them at the close of this part of the event as well. And then that's it. So please enjoy the posters. And in addition to the awesome posters today, there's also a happy hour at the same time. So enjoy yourself during that as well. Thank you all.